We will now take a look at a transduction pathway. A transduction pathway is often referred to as a relay race or a cascade, and it typically involves a series of protein phosphorylation, which means that an inorganic phosphate group will be added to one protein that will activate it, and this will typically activate another or transfer another phosphate group to another protein to activate it, and this will continue on for a series of steps. These pathways usually involve a series of kinases because a kinase is the enzyme responsible for adding phosphate to the proteins. These relays can end with a cellular response. This cellular response can be a specific activated enzyme. It could also involve the production of a secondary messenger such as calcium or cyclic AMP or CAMP for short. And it could also involve activating a transcription factor. Remember, a transcription factor, once it's active, will go into the nucleus to affect gene transcription. These transcription factors can turn on or off genes. They can also up or down regulate gene transcription, which means that they can make a gene be transcribed more or less. A transduction pathway is usually turned off by what we call phosphatases. Phosphatases are once again enzymes, but in this case, they remove phosphate groups from proteins. So the balance between kinases, adding phosphate to proteins, and phosphatases, which remove these phosphate groups, we can control these transduction pathways. We are now going to take a look at an example of one of these pathways. We are now going to take a look at an example of a transduction cascade. Here we have a hydrophilic signaling molecule binding to its receptor in the cell membrane. This will activate a relay molecule that will help pass this message into the interior of the cell. In this example here, we have an inactive PK1 or protein kinase 1. This will become active once the relay molecule becomes active to form an activated form of PK1. Protein kinase 1, remember, is a kinase, and a kinase is responsible for adding phosphate groups onto other proteins. In this case, the target protein of PK1 is protein kinase 2. So PK1 will phosphorylate PK2 and cause it to become active. Notice that if we wanted to inactivate PK2, we would simply remove that phosphate group. However, let's consider an active protein kinase 2. Once again, protein kinase 2 is a kinase, so it is going to phosphorylate another protein. And we will see our target protein at the end of this chain go from the inactive to the active form. This will then cause a cellular response. Once again, if we wanted to inactivate our target protein, we would simply remove that phosphate group. This would usually be done by a phosphatase. In our hydrophilic pathways, we typically have the cellular response involving the production of second or secondary messengers. So what exactly are these molecules? Secondary messengers are small, non-protein-based, hydrophilic or water-soluble molecules or ions. It is important to remember that these secondary messengers are not needed for hydrophobic ligands. They are not involved in those pathways at all. They are specific for hydrophilic ligands. They're responsible for creating and releasing a signal within the cell. This helps to spread the signal throughout the cytosol of the cell to get a very fast and quick response. Common examples of secondary messengers are CAMP, CAMP, or cyclic AMP, cyclic GMP, calcium, and IP3. We are going to take a look at the role of cyclic AMP as a secondary messenger. Cyclic AMP is produced by a plasma membrane enzyme called adenylyl cyclase. Adenylyl cyclase will take ATP, which is adenosine triphosphate, in the linear form. It will remove two of the phosphate groups, and it will create cyclic AMP, cyclic adenosine monophosphate. Cyclic AMP is an important secondary messenger. One of its main target enzymes is protein kinase A. Protein kinase A, once activated, will begin a protein phosphorylation cascade. If you want to shut down the pathway, you would activate phosphodiesterase. Phosphodiesterase specifically targets cyclic AMP and it activates it, meaning that it converts it back to the linear form. So when we have AMP in the cell, it is not considered to be a secondary messenger. It's only a secondary messenger when it is present in the cyclic form. Note in the diagram below the difference between the linear and cyclic structures. It is also important to note that there is a balance between G proteins and how they act on this plasma membrane-bound enzyme adenylyl cyclase. Some G proteins will act to activate adenylyl cyclase, thus producing cyclic AMP. However, some G proteins will work to inhibit adenylyl cyclase, thus preventing the production of cAMP. This will help to fine-tune a cell's response. This slide depicts an example of cyclic AMP and its role in the cell. Once again, we have the involvement of a membrane-bound receptor. In this case, it is a G protein coupled receptor. We have a signaling molecule in this case, such as epinephrine. Once again, epinephrine is a hydrophilic ligand. So the ligand would be binding to the G protein coupled receptor. This would activate the receptor and thus activate the G protein, which we see here in its active form because it is bound to a GTP molecule. This active G protein would slide along the membrane and activate, in this case, adenylyl cyclase. Adenylyl cyclase would take ATP molecules and change it into cyclic AMP. This is the production of the secondary messenger. This cyclic AMP will in turn activate protein kinase A. Protein kinase A would then cause a phosphorylation cascade to occur in the cell to then cause a cellular response. We are now going to take a look at two other secondary messengers, calcium and IP3. In this type of pathway, we have the involvement of phospholipase C. Phospholipase C is also another enzyme like adenylyl cyclase that is present in the plasma membrane for these signaling pathways. It will be activated in response to a signal. Once phospholipase C becomes activated, it will cleave PIP2, which is a plasma membrane lipid. When PIP2 is broken, it will form two molecules, IP3 and DAG, inositol triphosphate and diacylglycerol. These are two different secondary messengers. Diacylglycerol will remain near the membrane and it will cause an ion channel to open to allow for more calcium to pass through the membrane and to enter into the cell. IP3 is important because it will leave the membrane, it will bind to the smooth endoplasmic reticulum, and if you remember from our previous unit looking at organelles, one of the roles of the smooth endoplasmic reticulum is for calcium storage. When IP3 binds to the smooth endoplasmic reticulum, it will allow the smooth ER to release these calcium ions into the cytosol. Thus, we have a twofold increase in calcium concentration in the cell. One, through the use of DAG to open up the calcium channels, and two, through the opening of the channels on the SER to allow for stored calcium to also enter into the cytosol. Calcium itself is an important secondary messenger that can be used to activate many proteins in one or more signaling pathways. Here we're going to take a look at this specific pathway. Once again, we have a signal molecule that is considered to be hydrophilic. It will bind to the G protein coupled receptor. It will activate the G protein. As we can see here, the G protein is in the active form with GTP attached to it. The activated G protein will slide along the membrane to activate phospholipase C. Phospholipase C will target PIP2. We can see that PIP2 is simply a modified phospholipid that is present in the cell membrane. It will cleave apart this phospholipid to release DAG and IP3. We can see that DAG stays in the membrane, and remember, DAG would then bind to a calcium channel to allow for an influx of calcium across the membrane into the cell. We do not see that in this diagram, but that would be the role of that DAG molecule at the top right of this image. IP3 then is the remainder part of PIP2, and this IP3 will then be targeted to the smooth ER. Once it binds to the calcium channels on the smooth ER, it will open up these channels and allow for the stored calcium within the smooth ER to be released into the cytosol. Remember, calcium is also a secondary messenger, so it will help to activate various proteins within the cell to cause a cellular response. You might want to ask yourself, why are these transduction pathways present? What is their purpose? So they have many purposes within the cell. One of them is to amplify the signal. This means that the binding of one signal molecule could actually generate millions of secondary messengers, and those messengers could activate millions of other enzymes. So by amplifying the signal, once again, we can spread the signal to the rest of the cell very quickly. These transduction pathways can also help to organize and coordinate different cellular responses. There's many ways to fine-tune a pathway by activating or deactivating enzymes by producing or not producing secondary messengers. And so by having this level of organization, you can have crosstalk between pathways to ensure that the cell response is optimized. For instance, if we have cellular metabolism occurring and we have aerobic respiration, we've already seen that we can use fats, we can use glucose, and we can use proteins as energy sources for aerobic respiration. So if you have a large amount of glucose available to the cell, you would want to activate the enzymes for glycolysis and for metabolizing glucose molecules, and you would want to shut down the pathways for fat metabolism as well as protein metabolism. And this is one of the ways in which the cell can do that. Another important use for transduction pathways is to allow for different responses. This is important because one signal molecule can cause different responses in different tissue types and different cell types. This is because there are going to be specific receptors that are going to be either present in certain tissues or present at certain times. And there's also going to be specific pathways within the cell to cause a unique response. This slide here gives you an overview of the concept of amplification. In the reception phase, we simply have one molecule of epinephrine binding to its G protein-coupled receptor. If we take a look at the overall response, we can see that we have glycogen, which is going to be releasing many glucose molecules, in fact, 10 to the 8 molecules. This is done in response to the binding of one epinephrine molecule. So let's see the transduction pathway to follow how this occurs. At the beginning of this pathway, we have an inactive G protein. So the binding of one molecule of epinephrine will activate 10 to the 2 molecules of the G protein. This will cause the activation of adenylyl cyclase in a 1-to-1 ratio. So if we have 10 to the 2 G proteins activated, we will also have 10 to the 2 adenylyl cyclase activated as well. Adenylyl cyclase, remember, is responsible for converting ATP into the secondary messenger cyclic AMP. This does this at an amplified rate. So for 10 to the 2, or for every 100 adenylyl cyclase enzymes that are activated, we have the production of 10 to the 4 cyclic AMP molecules. These will each activate their own protein kinase A enzyme, once again in a 1-to-1 ratio. So we have 10 to the 4 enzymes of protein kinase A activated. Protein kinase A in this pathway will target the activation of phosphorylase kinase, and this is an amplified response at 10 to the 5. And this would then in turn activate inactive glycogen phosphorylase to the active form. And this would be amplified, and now we have 10 to the 6 activated glycogen phosphorylase enzymes. The role of glycogen phosphorylase is to break apart glycogen into glucose molecules. This would be our targeted cellular response. So we can see that the binding of one simple signal molecule can cause the activation of hundreds of thousands of molecules. This slide depicts an example of cellular coordination between pathways. If we take a look at the first part of this diagram, we have cell B, where the binding of one signal molecule will activate a relay molecule that will then activate two different pathways in the cell to cause two different cellular responses. Conversely, in cell C, we have an example of crosstalk, such that we have two different signal molecules binding, and the second binding of the secondary molecule, signal molecule, will cause the activation or inhibition of the pathway involved in the cellular response. So this allows for coordination between pathways. And finally, here we've got an example of multiple responses that you can have from the binding of a similar signal molecule. Here we've got one signal molecule causing 4 different responses within 3 different cell types. In the first diagram, in cell A, we have the binding of the signal molecule causing the production of relay molecules to cause a cellular response. In cell B, we have a similar binding of the same molecule. However, in this case, it will activate 2 different pathways for 2 different cellular responses. And finally, in the third cell, cell D, we have the binding of the same molecule. However, it will cause a different cellular response.